This is Med School Radio. We're looking at the immunology chapter of First Aid 2020. This time we're looking at complement. So we're going to look at complement, but first, before we talk about complement, let's say a few things about it. Remember that it is a system of hepatically, made in the liver, synthesized plasma proteins that play a role in innate immunity and inflammation. The membrane attack complex, or MAC, defends against gram-negative bacteria. The CH50 test is used to screen for activation of the classical complement pathway. So let's review the activation pathways. First, the classic. This is IgG or IgM mediated. So remember, GM makes classic cars. Next, the alternative pathway, microbe surface molecules. And next, lectin, the lectin pathway, mannose or other sugars on microbe surface. Next is the functions. So C3b is for opsonization. C3a, C4a, C5a, anaphylaxis. C5a, neutrophil chemotaxis. And C5b through 9 is the MAC, membrane attack complex, cytolysis. So remember that C3b binds to lipopolysaccharides on bacteria. This is for opsonization. C3b opsonization, C3b binds to lipopolysaccharides on bacteria. An important part of that section is C5b through 9, MAC cytolysis. This comes up a lot in questions. Next, opsonins. C3b and IgG are the two primary opsonins in bacterial defense. They enhance phagocytosis. C3b also helps clear immune complexes. So remember, the meaning of opsonin, which is Greek, is to prepare for eating. Next, inhibitors. Decay accelerating factor, or DAF, aka CD55, and C1 esterase inhibitor help prevent complement activation on self cells, for example, red blood cells. And then at the bottom of the page we have the figure showing the different pathways, the alternative, the lectin, and the classic. Just to remind you, the alternative pathway, spontaneous and microbial surfaces, amplifies generation of C3b. The lectin is for microbial surfaces, for example, mannose, and the classic, antigen-antibody complexes. Thank you very much.